Dear student, welcome you back to my YouTube channel Learning Island. Today I am going to discuss the topics heredity from class 10 chapter 9. Here I will discuss two main topics which are very relevant in your term 2 examinations point of view. These are Mendel's monohybrid cross experiment and dihybrid cross experiments. You know, Gregor John Mendel is the father of the heredity. He had done these two experiments and shared the results with us. Now, before going to explain those experiments, I will have to share some definition with you and make you understand what are those definitions, because these definitions are very relevant with these two experiments. So let us find what are those definitions. First of all, what is heredity? You all know the transfer of characters from parents to offspring is known as the heredity, and the process through which the traits and characters pass from one generation to another is called the inheritance. Next, what is allele? An allele is one of two or more versions of a gene. An individual inherits two alleles for each gene, one from each parents. If the two alleles are the same, then the individuals is homozygous for the gene, and if the alleles are different, the individuals is heterozygous. So here you see in this picture in the left side, both the gene are same categories, capital A and capital A, which are dominant characters. Both are same, that's why it is called homozygous. And in the second, one chromosome is capital B, one is small b. So one is dominant and another is recessive. That's why it is called heterogeneous. Now I will discuss what is variation. So variation, it refers to the difference in the characters or the traits among the individuals of the species. So here you see at the time of why the variation actually happen. At the time of chromosome mutation, these types of two chromosomes are there. This is like spiral shapes, and when the mutation happens, that is the cross reproduction, then some errors happens in DNA, which results the variations. So this is called the variation. Here you see in the list of a giraffe, this is brown, this is yellow, this is black, brown black mix. So this all kinds of variations are there in in our human life. You will not find any similarities between the parents and the offspring or the babies because of these variations only. So if there is no variation, then the no upgrades or any development or any evolution would have been happen. So the variation is most important in the generation to generation for the development of the generations. Now I can go to the next thing, that is the monohybrid experiment. So let me go to the next step. This is Mendel's monohybrid experiment. So here you see I am discussing what is monohybrid experiment. Why it is monohybrid? Because in this case the Mendel had taken only one characters from male and another pure character from female. That is only one character has been taken. That's why it is called monohybrid. So Mendel had taken pure tall pea plant as male, and he has symbolized it by capital T, capital T, which are dominant character, and also taken female as pure short seed of pea plant and symbolized it as small t. So this is called pea, and to make it hybridizations, he take one chromosome which is called gamete, and from here also taken one chromosome which is called gamete. So this is p gamete stage. This is p stage parent. This gametes he made cross pollination between this, and after cross pollination he got small t capital T, that is mixed pea plant. Now this is called F1 generation. So from this F1 generation he got the mixed pea plant, and he made again among these self pollinations, because these are the same species, and he made self pollination. So in this stage he got four types of plants. One is capital T, another is small t, capital T, small t, and small t, small t. So here you see, after self pollination with the with the plants got in the F1 generation, he got four types of plant. So here you see, if I see from outside, here we will get three types of tall, because capital T is the dominant characteristics, so small t will not show here because this is the recessive characteristics. So from outside obviously we can see only the tall characteristics. So three types of talls, and one is pure short. So you can say the phenotypic ratio, this is called phenotypic ratio 1 3 is to 1. And if I go inside, if I show the gene, that allele, that is the gene here is all are capital T, that is pure tall. This is capital T and small t. This is the mixed. This is mixed. These two are mixed, and one is pure short, one is pure tall. So the genotypic ratio would be 1 is to 2 is to 1. So what is phenotypic and what is genotypic, I will discuss it clearly in the next slide. Now this is the figure of F2 generation. Mendel name it F2 generation. So this is the rough figure for your understanding. Now I am showing you the main figure, how it is look like. So this is the stage, parent stage, pure tall, pure short. Take the gametes, P gametes, then make the self cross pollinations. He got the mixed tall, and among these mixed tall he made again the self pollinations. He got the four types of plants, pure tall, which I have shown in the rough sketches. So here you see, this is overall figure is called the checkerboard, Mendel's monohybrid experiments checkerboard. Now to get this four types of plants, after making the cell pollination between these two gametes, uh, it is to find out the easily, the mathematician Punnett has made a mathematical calculations in a square form. This is called the Punnett square. Mr. Reginald Crondall Punnett, a mathematician who has invented this square to find out the offspring easily, the combinations of the offsprings. This is very easier calculations. So this is called the Punnett square. In your exam, this question is also asked. Write down the Punnett square for the monohybrid experiment. So this is the Punnett square. This is the Punnett square. So how it is obtained? Here you see this. These are the male gametes, capital T and small t. These are the female gametes, capital T, small t. So when you cross it, T multiply T, we will get the result here. TT. Again, capital T multiplied by small t, we get the capital T, small t. Again, from with this gamete, male and female, capital T, small t, again get capital T, small t, small t, small t, you get the small t, small t. So this is the way how to find out the offsprings get out of the gametes taken from the F1 generations. So this is the Punnett square. Now this is the phenotypic ratios and this is the genotypic ratios. So here the checkerboards in the left side and the Punnett square in the right side. So hope you have understand this monohybrid experiments very easily. Now I can go to the second stage. This is the Mendel's. Okay, in this experiments, few topics or few definitions I have explained, that phenotypic, genotypic, and dominant recessive. So what are they actually? These are, what is dominant allele? You go through yourselves. And what is recessive? You go through please. And what is phenotypic? Because these all types of definitions are there in your books, but I am explaining a little bit because so that you can understand the experiments easily. So what is dominant allele? So dominant means the characters which is which is you can see from outside. This is the dominant, and the recessive means the characteristic which is not shown, but in the gene there is that characteristic. Suppose your from your grandfather has a curly hair, but you or your brother is not that kind of curly hair. What happened? This is the phenotypic, that is the curly hair is not showing in you, but this characters or this gene is lying within you, is in your gene. So the characters or the traits which are not shown, you know, from the external side or outside, this is called the recessive actually. So, and the phenotype is the ratio which has been shown in the first experiment, that is 3:1, and in genotypic it is 1:2:1. So let us go to the his dihybrid experiment. In dihybrid experiment also he had taken pea plant. In this case he had taken two characters, that's why it is called dihybrid cross. So two characters are what? He had taken yellow, pure yellow and round, pure yellow and round, and pure green and wrinkled seed of pea plant. So yellow, pure yellow and round, he has symbolized those by capital Y and capital R, capital R. In this case he has symbolized those small y, smaller, smaller. So if we take the gamete from this, we will get capital Y smaller, and from this small y smaller. So these are called P gametes. P gametes. So when he made cross pollination among this, cross pollination, he got capital Y, small y, small y, capital R, small r. This is mixed, but from outside phenotypic it is showing that this is a yellow and round seed, but this is actually mixed. yellow, green, round, wrinkled. So this is the F1 generation. F1 generation stage. So see, there is a similarity between the monohybrid and dihybrid cross. There is also all these steps: parents, P gametes, F1 generations. Now again from this F1 generation, he had taken these two mixed plant of pea plant and made, self pollinations. He made self pollination again between them, like previous in the monohybrids cross. Self, he made self pollination. So during self pollinations, if we take gametes from here, we will get like this, gamete. How to take the gamete? So gamete will be like this: YR, 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 YR. That is capital Y, capital R, capital Y, capital Y, small r, small y, capital R, small y, small r. Here also get capital Y, small r, because like this: capital Y, small r, capital R, again capital Y, smaller. How? This and this. Then small y, capital R. How? This and this. Then small y, smaller. How? This and this. Now if they make the self pollinations among these, so to make it understand properly, we will show you through the checkerboard. Now let us the steps which have been shown right now in proper way, because this was the rough sketch for your understanding. Now see, these are the steps which I have explained right now. So you just pause the video and write it down, or you can take the screenshot also. This is the checkerboard of second, that is dihybrid experiment of Mendel's. Here you see the steps are similar like the monohybrid cross. Parent is the first step. This is the parents, and from the parent take the gamete. So P gamete stage, then make the cross pollinations. These are the F1 generations, and from F1 generation we have taken the gametes for F2 generations. So let us go to the F2 generations checkerboard. This is the checkerboard. These are the gametes are placed over here. See, gametes are placed over here. Now I will show how with the help of Punnett square we can make the offspring. So here you see, if I cross these and these are the all, these all are the male gametes, these are the female gametes. So if I make the cross among these, I will get R, capital R, capital R, and capital Y, capital Y. Here this and this multiply, will get capital R, capital R, capital R, capital Y, small y. And here capital R, small r, capital Y, capital Y. Here this and this we get capital R, small r, capital Y, small y. So in this way we can get the other like this. You see, in this way we can get the other. You just try your best to get the others. So this here is some, I think this is some mistake over here. you can cut it down, you can write it down like this: capital R, capital R, small r, and capital Y, small y. So in this way, in this these are the, so if I see, there is as I had already told you that the capital R and capital Y are the dominant characteristics. So wherever you find these capital and capital R and capital Y, then it is, so this is round and yellow. This is also round and yellow because capital R and capital Y are there. This also round and yellow. This also round and yellow because capital R, capital Y are there. This is also round and yellow. This is 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 round. So total 1, 2, 3, 4, 5, 6, 7, 8, 9. Again numbering 1, 1, 2, 3, 4, 5, 6, 7, 8, 9. So total 9. So pure yellow and round, total 9 number. Again if I see this round and green, this is round and green, because you see capital R but small y is the green, capital is the round. So round and green, so round and green are 3. Again this is the wrinkled and yellow. So wrinkled yellow wrinkled yellow are 3, and 1 is left. This is green and wrinkled. So green wrinkled equal to 1. So these are the phenotypic ratio. So this is, you can say, 9:3:1. Now what is the genotypic ratio? So genotypic ratio is, if I go inside and see check the gene level, then I will get this ratio. This is 1:2:2:2:2:4:1:2 2:1:2:1. So how I got this ratio, I will show you this in the next slide. So let us go to the next slide to understand the genotypic ratio. So I think you have understand the phenotypic ratio. So please write it down or pause the video and note it down, and also you can take the screenshot. So let us go to this slide to understand the genotypic ratio. Here you see from the, if I go back, from the among these 9, out of these 9, if I take the genotypic symbol, among these 9, this is the phenotypic first 9. This is only the 1. So this is 1, and these are similar because capital R, capital small, capital Y, small y, capital small, capital Y, small y. So I can give this number 2. So 1:2. Now if I check this one also, capital R smaller, capital Y, small y, capital R smaller, capital Y, capital Y. So this is also 2 numbers. So 1:2:2:2. Now these are same. 4, there are 4 numbers same. So 4, 4, 4, 4. So this ratio becomes this 9 can be split as 1:2, 2:2, 2:4. You see 4 plus 2 plus 2 plus 1 equal to 9. So 9 phenotypic, in terms of genotypic, it is 1:2:2:4. Again if I come to this 3, these 3 are, one is this, one is this, and one is this. So here also this is the similar single, and these two are similar. So I can write again from phenotypic. If I come to the genotypic, this 3 becomes 1:2. Again from another 3, these and these are 3. So this is the single and these two are similar. Again from 3 we get 1:3. 1:2, sorry, 1:2. And now rest 1 is 1. So this is 1. That's why the ratios comes 1:2, 2:2, 2:4, 4:1:2, 2:1:2, 1:2:1. So now you let us understand it more clearly. Like this I am showing you. See, this is 1, so in the ratio it has come 1. You see, again this is only one characteristic. Now there are 2, so again 2, so 1:2, then again 2, 1:2. Again these 4 are the same characteristics to the 4. Again this is the only one characters genotypically, so again 1. Again these two are similar, so again 2. Again this is the single, so 1. Again these two are similar, this is 2, and rest is 1. So again I am showing you, going back, genotypic ratio 1, so 1. Again 2 are similar, so 2, 1:2. Again more 2 are similar, so 1:2:2. More 4 are similar, so 1:2:4. Then again 1, so 1. Again 2 are similar, so 2. Again 1 is similar, so 1. Again 2 are similar, so 2. Last one is 1. So finally we have got 1:2, 2:2, 2:2, 4:1:2, 2:1, 2:1. If we add all this, we will get 9:3, 3:1. So total 16 are there. So this is called the Punnett square. So I think this video remain very helpful to you, and now you can easily understand what is Punnett square for dihybrid and monohybrid cross, experiment of the Gregor John Mendel, and also what is checkerboard, and how to write the checkerboard step by step. So thank you all for being with me throughout the videos. So hope you will do well in the term 2 examinations. So thank you very much again. We will see you in my next video. Thank you. Bye bye.